So good morning, everyone. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respects to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. My name is Maria Elena, and I'll be your chair for today's seminar. I am a PhD student of the center at the Biotechnology Institute of the Uni Melbourne node. And I would like to start with a few housekeeping rules. So first and foremost, our seminar today will be recorded, and you will be able to find these and our previous recordings to the CCMMP website. Letting you all know that the presentation of our speaker will be 45 minutes, followed by a 10 to 15 minutes Q&A session. So please leave your questions to the end of the seminar, either by raising your hand, or if you feel free, just type them in the chat box and we can address them later. And please make sure that your microphone is muted throughout the presentation. Our presenter for today's seminar is Associate Professor Megan Maher. Megan was awarded her PhD in inorganic chemistry from the University of Melbourne, and then proceeded to 7 years of postdoctoral studies at the University of Sydney. She became a team leader at the membrane protein crystallography lab at Imperial College in London, and she returned back to Australia in 2008 to the Centenary Institute in Sydney, and then to La Trobe University. In 2018, she was appointed to the position of Associate Professor. And 1 year later, she joined the School of Chemistry and Biotechnology Institute at the Uni Melbourne. Professor Maher has been awarded with prestigious awards and fellowships throughout her career, including the Georgina Sweet Award for Women in Quantitative Biomedical Science and the ARC Future Fellowship. Professor Maher's current research focuses on investigating the role of trace metals in biological systems, with a particular focus on the structural biology of metal proteins and integral membrane protein transporters. Her presentation today has the title Structural Insights into Manganese Transport Across Membranes. And a warm welcome to you, Professor. We're really lucky to have you today. The screen is all yours.
Thanks very much, and thanks very much for that really kind introduction. Also for the invitation to speak, actually. I've been really enjoying this series of seminars, so thanks for including me. So today I'd like to tell you about some recent, relatively recent research from my lab that I thought, given I'm a new member of the centre relatively recently, I'd use the opportunity to introduce the research that we focus on in my lab. So as mentioned, I'm an inorganic chemist by training. My lab's in the Bio21, but we're part of the School of Chemistry. And the reason, and this has all come together to really give me my research focus, which is looking at the role of trace elements in biological systems. Now, if you have a look at the back of this bottle of vitamins and minerals that you might all be familiar with, that you might take as a supplement, you'll notice on the back here there's quite a number of transition metals that are included in this cocktail, and they include zinc, copper, manganese, and molybdenum. And this is because these trace elements, which are transition metals, are essential for all forms of biological life. You can see I've highlighted them here on the periodic table, and these are the types of elements that are essential across all biological systems, and they even include rather obscure sounding elements such as molybdenum and tungsten. So as I said, they're required for all forms of biological life. We know that about 30% of proteins bind these transition metals, and about 50% of enzymes require metals for activity. And here are some of the essential biological processes that use these transition metals. DNA transcription requires iron and zinc. Photosynthesis in plants requires magnesium, manganese, iron and copper. And respiration in mammals, including us, requires iron and copper. So we've known about the requirement for these trace elements for a really long time now, but when I was an undergraduate, which was probably a really long time ago also, we were taught that the interior of cells were really metal soups, and the metals just floated around randomly, ready to be picked up by the systems that required them. And there was a bit of a revolution of thinking in the late 1990s that revealed that this was not the case, that these metals are actually very, very tightly controlled in biological systems. And the reason for that is that they're not only essential, but they're also toxic if dysregulated. And so we refer repeatedly to metal homeostasis as being a situation in the cell where metals are very tightly regulated and used for their essential functions. So what we know now is that in all cells, there are protein-based systems that control metal availability and function. They consist of integral membrane proteins, shown here, which are transporters that control the uptake of these trace elements. They also act in efflux if the concentration is too high within the cell. They include metallochaperone proteins. These are soluble proteins that bind the transition elements and deliver them to the systems that require them. And they also include metalloregulator proteins, which are mostly transcription factors that turn the expression on and off of these other systems in response to fluctuations of concentrations of metals in the cell. So when this homeostasis goes wrong, there are diseases that are associated with that. And a really obvious one is anemia and hemochromatosis, anemia being an iron deficiency in humans and hemochromatosis being an iron overload. There are corresponding diseases that relate to copper. So Menkes disease is a copper deficiency, Wilson's disease being a copper overload disease. But there's also metal homeostasis is linked to neurological diseases. These include prion diseases, Parkinson's, Alzheimer's, and ALS. And all of these diseases have some component of metal dysregulation in their pathologies. Finally, a more recent field of research has revealed the role of metals in infection and immunity, and that's what I'll talk about today. So what's become apparent is that at the host pathogen interface, the regulation of transition metals is used as an innate immune response, and another term for this is called nutritional immunity. So this is a bit of a tug of war between the pathogen, for example, a bacterial infection, and the host, for example, us. So in response to such an infection, iron is sequestered away from the infection. It's tied up in proteins such as transferrin and ferritin. And this is an effort to starve the pathogen of this essential element. Iron is required for respiration amongst other functions. And it's estimated that something like a bacterium like E. coli requires about 100 times within the cell, 100 times the iron concentration that's actually available in the environment. So it has a lot of overlapping processes to accumulate iron. And so the host response is to sequester this away in order to basically starve the organism of this nutritional component. On the flip side, copper and zinc can be used as bactericidal agents. So we know, for example, in macrophages, the copper and zinc transporters are upregulated in the cell membrane. They pump these metals toward the invading pathogen, and this is in an effort to poison the infection, essentially, as these have bactericidal functions. So the organism we've been working on for a while now is Streptococcus pneumoniae, and this is a close collaboration with Chris McDevitt from the Doherty Institute. Streptococcus pneumoniae causes inner ear infections and pneumonia. Until COVID, it was recognised as the leading infectious killer, basically, of children under 5 years of age worldwide. We have vaccines against Streptococcus pneumoniae. They target a handful of the serotypes. Although they're very effective against those serotypes, what's happening is that other serotypes that are not covered by the vaccine are emerging in the population. We also know that Streptococcus pneumoniae, there's increasing prevalence of antibacterial resistance or antibiotic resistance. And this has been listed by the World Health Organization as an organism of concern in terms of that antibiotic resistance. So we need new tools to fight Streptococcus pneumoniae infections, and that's what our focus is in this project. And to do that, we've been looking at the metal requirements of the organism to see if we can harness that as a new strategy for antibacterial design. So in Chris's lab and in the labs of others, it's been shown that this organism has an essential requirement for manganese. It's also been shown that although zinc is essential, it's also toxic when in certain concentrations. The toxicity of zinc comes from a number of factors. It prevents the acquisition of essential trace elements such as manganese. It leads to oxidative stress and it inhibits cell wall biosynthesis. In humans with zinc deficiency, we know they have trouble fighting Streptococcus pneumoniae infection. So we're asking whether this zinc toxicity can be harnessed for antibiotic design. So this is a bit of an illustration of some of those points, data coming from Chris's lab. In this case, this is data from a mouse model of infection with Streptococcus pneumoniae. And in response to infection, the zinc concentration in different infectious niches has been measured. And you can see in response to infection, the zinc concentration increases quite dramatically. This is this innate immune response that I spoke about earlier. The other point is that zinc is toxic to this organism. So this D39 is a laboratory strain of Streptococcus pneumoniae, and you can see when it's grown in conditions of increasing concentrations of zinc, the zinc inhibits its growth. This can be augmented by knocking out one of the zinc efflux pathways. This is an integral membrane protein transporter that effluxes zinc from the cytoplasm. And you can see if we knock that out, then the organism becomes even more sensitive to zinc. So what we'd like to do in collaboration with Chris is understand the molecular mechanisms behind this zinc toxicity and how Streptococcus pneumoniae might resist this zinc toxicity. And to do that, we're going to define the protein systems that are impacted by or that respond to zinc, solve their structures, define their functions, and identify possible targets for antibacterial design. So what I'll talk about today is a focus on the integral membrane protein transporters that control zinc uptake, efflux, and also that are impacted by zinc. So these are the transporters I'll be talking about. You'll see they have terrible names in terms of all sorts of letters, but I'll try and keep that to a minimum because it becomes very confusing. But these are the three transporters we've been focusing on in trying to understand this zinc toxicity. So the first one is an ABC transporter. It's ADCCBA. It's got a typical ABC transporter structure. It has two nucleotide binding domains, two transmembrane domains that sit in the membrane, and also includes a solute binding protein, ADCA, which binds zinc and delivers it to the transporter for transport across the membrane. So this transporter brings zinc into the cell. When zinc accumulates to concentrations that are too high, the zinc is effluxed out of the cell via this transporter here, CZCD. This is the one that was knocked out in that strain of Streptococcus pneumoniae I showed you before. This is a cation diffusion facilitator protein. It uses the proton motive force for powering zinc efflux out of the cell. The main focus of the talk today will be this transporter here. This is a manganese specific transporter called PsaBCA. It's got the same overall structure as the one I've already mentioned. Nucleotide binding domains that sit in the membrane, nucleotide binding domains that sit in the cytoplasm, transmembrane domains in the membrane, and a solute binding protein that binds manganese and delivers it to the transporter for transport across the membrane. So PsaBC belongs to this type 2 importer family, and the most well-studied example of that family is a vitamin B12 transporter that has been worked on quite heavily by Caspar Locker's lab. And they have solved the structure of the transporter in a number of conformations under different conditions and in complex with its solute binding protein. So one of those structures is shown here, and you can see the overall topology of the two nucleotide binding domains in the cytoplasm the transmembrane domains in the membrane, and the solute binding protein is associating here with the transmembrane domains for delivery of the cargo. So the cargo is translocated between the transmembrane domains here, and it's bound up by this solute binding protein that delivers it and then interacts with the transporter for release and transport across the membrane. So the work that the Locker Lab have done, they've come up with, I guess, a general philosophy about how these transporters work. And their proposal is that the specificity for the substrate or for the component or the cargo that's going to be transported comes primarily from this solute binding protein, that the solute binding protein binds vitamin B12 in this case very specifically, and then when it docks with the transporter, it releases it for translocation. Their proposal is that the substrate does not interact with the transporter at all. That's released and it basically slides through what they call a Teflon coated channel through the membrane. So this is going to be a little video to show you how ABC transporters work. They fluctuate between an outward open and an inward open conformation, and that is controlled by ATP hydrolysis. This protein I'll show you doesn't have a solute binding protein, but it shows you the general mechanism. So you can see it's outward open in this conformation. The cargo comes in, ATP is hydrolyzed in the nucleotide binding domains, and that causes a switch to an inward open conformation that allows the cargo to go through the membrane. Okay, so I mentioned before that one mechanism of zinc toxicity in this organism is that it prevents acquisition of essential trace elements, and as I said, it prevents acquisition of zinc. So this is all through zinc's action on this PsaBC transporter. So I've represented the transporter again here, and in this case I'm showing two different conformations of the transporter. We have the outward open conformation that would dock with the solute binding protein to deliver the manganese to the transporter, and then once it drops in, ATP hydrolysis causes the nucleotide binding domains to separate and allows the cargo to go through the membrane. So Chris has done quite a lot of work on this solute binding protein, which is called PsaA, and he's done a lot of structural work in collaboration with Boston Kobe's lab at the University of Queensland in trying to understand why zinc prevents manganese uptake. And what they found is that both zinc and manganese bind to the solute binding protein with approximately the same affinity. But when zinc binds to this solute binding protein, it's actually basically locks it. It's a very stable complex, and then solute binding protein cannot release zinc into the transporter. So basically when there's too much zinc around, it ties up this solute binding protein with zinc bound, which can't be released, and in that way prevents manganese uptake. So zinc toxicity leads to manganese starvation. And the reason for that is basically comes down to coordination chemistry. I'm going to show you a morph here of the PsaA protein. This is in the metal-free conformation, and then we're going to bind manganese and zinc. So this is the free conformation. You can see it has these two lobes. When the metal comes in this case manganese, these amino acid ligands here rearrange to coordinate the metal, and these two lobes close down on the substrate. And then as it switches to zinc, it'll become grey. You can see it tightens up even further, and the unwinding of this alpha helix along the back allows that to happen. When zinc's bound, it's bound in an almost ideal tetrahedral coordination, which is very stable, as I mentioned, and so the zinc cannot be released to the transporter. Manganese, in contrast, is bound in a bit of a distorted octahedral coordination geometry, which is less stable, and so when this docks with the top of the transporter, the manganese can be released for translocation. Okay, so I thought since there's some students in the audience today, I'd just give a couple of slides on our approach to studying integral membrane proteins in my lab. We have a pretty well-established workflow now aimed at structural determination of these transporters. In this case, I've given an example of what we would do for crystallization or to solve the structure by X-ray crystallography. You'd all be familiar with the requirement to generate recombinant protein expression and purification. In our case, we predominantly take that protein to crystallization trials, collect X-ray diffraction data and solve the structure. And this is also appropriate for cryo-EM, and we've started doing a little bit of that very recently. What we find is that one of the major bottlenecks is this point here, where we need to overexpress and purify the protein. For crystallography, we need pretty relatively large quantities of pure protein, and it needs to be very pure and very stable. So what we do is we've built on the work of David Drew, who I worked with at Imperial College and now who's at Stockholm University. And he's developed a workflow based on GFP tagging of the target membrane proteins. So in the case of PsaBC, we have PsaB open reading frame overlapping with the PsaC, which is the integral membrane protein component. And we tag that at the C terminus with GFP, which has a polyhistidine tag for purification. And we have an intervening protease cleavage site, which is either TEV or 3C. The advantage of the GFP tagging is it allows us to do a lot of expressions trials from whole cells to optimize the expression levels. We can screen detergents for both protein for extraction and purification using the GFP tag. In expression systems such as yeast or insect cells or mammalian cells, we can use the GFP to check that the protein is targeted to the membrane, and we can use STS page to locate our proteins. So this is a bit of an illustration of this from David's paper. So you can see here, these are expression trials for GFP tagged membrane protein. In this case, they've used Cumazi staining to try and identify the overexpressed protein. You can see that's very difficult. But if we use the GFP fluorescence, it's very easy to find our overexpressed protein, and we can check that it's full length, which is really important. We use fluorescent size exclusion chromatography, where we use membrane extracts and we pass them down a gel filtration column, and we use the fluorescence to tell us where the fusion protein is eluting, and we can therefore screen a whole host of detergents and look at the integrity of the protein, whether it's aggregated, whether there's a lot of free GFP, etc., using that profile. In my lab, we have an inline fluorescence detector connected to the size exclusion chromatography, so we can do this very readily. And as I said, if we're using expression hosts such as yeast, we can check that the protein goes to the correct membrane. So this is a bit of a snapshot of the PsaBC purification. You can see it eluting from the nickel resin here and in the dialysis bag. So it's very easy for us to track that we're isolating our protein using this GFP. So we did all that for PsaBC. We tagged it with GFP, and we were able to overexpress and purify the protein that was very well behaved, very stable in solution. And we were able to grow some beautiful crystals. You can see these lovely hexagonal looking crystals here, and collect diffraction data from them. But there's always a but. The diffraction data was not fantastic, to be honest, in terms of resolution or quality, in terms of diffraction, the statistics for the diffraction data. And more importantly, the space group of these crystals was triclinic, which meant we had to collect quite a lot of data to get a full data set, which is difficult given that membrane protein crystals suffer from radiation damage very easily. Well, it also meant that the search for heavy atom derivatives or even a selenomethionine derivative looking for the heavy atom sites would be very difficult using a triclinic space group. So at this point, we got a little bit stuck with this project, but we were very lucky to have a collaboration with Sowawata's group at Kyoto University, who had set up a pipeline for generating antibodies that target folded membrane proteins. So the approach they use is, in the case of PsaBC, to incorporate it into proteoliposomes and inject those proteoliposomes into the mouse. Once antibodies are identified, they're screened using an ELISA assay, as shown here. And the most important point of their workflow is to not only run the ELISA assay, but also to run a denatured dot plot in parallel. So what they look for are antibodies that light up in the ELISA that actually give no reaction in the dot plot. And what this indicates is that these antibodies are recognised as folded protein and not unfolded protein, which is what the dot plot recognises. And by doing this, we can identify antibodies that recognise the membrane protein when it's actually folded, and in that respect might be useful for co-crystallization. So those antibodies are cleaved down to antibody fragments or Fabs. And the idea here is that the Fab binds to the integral membrane protein and facilitates crystallization. So if you can imagine your membrane protein here as being encapsulated in a detergent micelle, you can see that in the case of many membrane proteins, not very much structure might protrude outside the micelle. And we require structure outside the micelle in order to make protein-protein interactions that scaffold the crystal formation. By binding Fabs to these parts of the structure that protrude outside the micelle, we increase the surface area of protein structure that's available to form crystal contacts, and so hopefully facilitate crystal formation. So the Iwata laboratory were able to supply us with 8 different Fabs, and we screened those in my lab by size exclusion chromatography. So here's PsaBC alone, eluted from size exclusion, and then we've incubated it with the Fab, and you can see a shift to higher molecular weight that indicates we have a PsaBC Fab complex. And we took those complexes and submitted them to crystallization trials. So when we did that, we grew one really enormous, beautiful crystal, and that was enough to solve our structure. So that's shown here. It was in a much better space group in terms of symmetry, and we could easily collect a full data set. And then because we had the Fabs attached to the protein, we are able to solve the structure by molecular replacement. So the molecular replacement used a model of the Fab in conjunction with a model of the nucleotide binding domain from a homologous transporter. And so this is the structure of PsaBC in complex with Fab. You can see here we have in blue the 2 transmembrane domains, and in pink the 2 nucleotide binding domains, and each of the nucleotide binding domains is associated with a molecule of Fab. We looked at the crystal packing, and just like we hoped, shown in this schematic where the Fabs would mediate the crystal contacts, we found that that was indeed the case. You can see the Fab here in red, and all the crystal contacts in the crystal are mediated by Fab-Fab interactions. So once we had that structure, we could return to our P1 crystals where we were able to collect a single full data set, and then use the structure of PsaBC to solve that structure by molecular replacement. So that structure is shown here. This is PsaBC alone in the absence of Fab. Again, you can see the 2 nucleotide binding domains in pink and the transmembrane domains in blue. Here I've overlaid the structure of the Fab complex with the PsaBC alone. I've just omitted the Fabs for clarity. And you can see that the structures are very, very similar. So there's no major structural changes due to the presence of the Fabs in one of those crystal forms. We collaborated with Megan Maher, who's now at the University of Queensland, who's done some MD simulations and sat PsaBC in the membrane. You can see that the membrane, unsurprisingly, encapsulates the entire transmembrane domain. There's very little structure protruding from the top of the membrane here, and the nucleotide binding domains lie in the cytoplasm. The other thing the MD showed was that the structure was pretty representative of what from the crystal was pretty representative of what it would look like in the membrane. There's no major changes once she modeled that into a membrane environment. So what does the structure of PsaBC tell us? Well, the first thing we can do is compare it to other transporters from this type 2 class. This one is the one I mentioned before from the Locker lab. This is a vitamin B12 transporter, and this one is a molybdate transporter. The other type 2 transporters solved from bacteria all transport really large cargo, vitamin B12, heme, and molybdate, whereas the PsaBC transports single manganese ions, which are tiny in comparison to the cargo that these transporters transport. And you can see even at this level, there are major differences between the structure of PsaBC and these other comparable transporters. One of the major differences is that PsaBC contains 9 transmembrane helices in comparison to these others that have 10. You can see this extracellular, little tiny extracellular helix here, which is at the N-terminus. In the case of these other 2 transported, that extends into a full additional transmembrane helix, which is the N-terminal helix. This makes the overall structure of PsaBC more compact. You can also see that the nucleotide binding domains have a really different sort of relative conformation to these other homologs here. We can see that in a bit more detail on this slide. So again, this is PsaBC compared to all the other type 2 transporters that have been solved to date. And you can see the compact packing of the transmembrane helices, particularly if we look down from the top of the membrane, looking through to the cytoplasm, you can see the packing is very, very tight compared to these. Interestingly, also, if we look also from that view, if we were looking down through the transporter and then down into the cytoplasm and looking at the nucleotide binding domains, you can see this difference in relative conformation of these domains. So in the case of these other transporters, the domains sit very close together and these Walker motifs line up very nicely. In this case, they've used a non-hydrolyzable ATP analog to really look at how those motifs align. But you can see even in the absence of ATP, they're aligned quite well compared to the ATP bound form. But in the case of PsaBC, they're very offset. So we think a major conformational change is required to bind ATP, and this would be a lateral change parallel to the membrane would be required to bring these 2 nucleotide binding domains together and then for ATP hydrolysis to occur. We've also looked at the translocation pathway through the transmembrane domains. This is basically a map of that cavity, and you can see that it's pinched off very, very tightly here. The diameter here is only 0.7 angstroms, and then it opens up as you go through the membrane. So this is an inward open conformation of the transporter. When the Locker lab were doing their work on the B12 transporter, they proposed this inward open conformation was unstable. We're now increasingly seeing this conformation in many, many structures of other type 2 transporters. And I think it's more indicating that this might be a resting state for the transporter, but we need to do more work to establish that. When it's pinched off so tightly here, this would prevent manganese efflux back through the transporter. Again, this transporter has to be very different to the others in that, as I said, it needs to transport something so tiny. Again, Megan O'Maher's lab has mapped where water is through the translocation pathway, and you can see that water is excluded as we go through the middle of the transmembrane domains here. So we propose that manganese comes in probably in a hydrated state, that water is stripped off as it passes through the transmembrane domains and then rehydrated as it exits at the bottom of the membrane. So this figure here is an electrostatic surface of the transporter, and I've cut away to look through the pathway through the membrane. And you can see in the middle of this pathway is a little cavity which is very negatively charged. So this is the pinch-off point I mentioned before, right before this cavity, and this cavity sits in the middle of the translocation pathway. And when we go and have a look at this cavity, we see that it's lined by a histidine residue and an aspartate residue from each of the monomers of the transmembrane domain. Now, these are typical metal coordination residues, and these are seen, histidine and aspartate residues appear in manganese-dependent enzymes. They also appear in other transporters such as zinc transporters and manganese transporters of the cation diffusion facilitator class, and have been characterized as important in facilitating metal transport through those transporters. It seems a common theme in transition metal transporters to have amino acid side chains that interact with the metal as they pass through. Another example is the CTR1 transporter, which is copper-1 transporter from higher organisms. In this case, methionine residues are actually used to coordinate the copper as it goes through the membrane. So we're proposing that this is essentially a metal sink within the translocation pathway, that these residues coordinate the manganese as it passes through, and this provides a driving force for manganese passing across the membrane. So to check that, Chris, in collaboration with bioinformaticians at the Doherty Institute, did a search of this motif against all the sequences of PsaBC type transporters, and found that these two residues are very, very highly conserved in other transporters that are most probably manganese transporters of the PsaBC type. And they could not only be identified in bacteria, but also in some higher organisms that actually do not have a solute binding protein. So we feel that these residues are absolutely essential for manganese translocation through the membrane. And so Chris tested that by basically in Streptococcus pneumoniae by knocking out the PsaC, which is the transmembrane domain, and then recomplementing with point mutants. So one point mutant where the histidine was taken out, and a second where the aspartate residue was substituted for an alanine. And you can see in the knockout and the 2 point mutants that the organism doesn't grow so well. And importantly, that manganese accumulation into the organism is attenuated by these mutations. So this is the wild type protein in the organism. The organism is able to accumulate manganese. When we knock out the whole transmembrane domain, it's no longer able to accumulate manganese to the same extent. And equally, if we just mutate one or the other of these residues, these also very significantly attenuates manganese accumulation. So this all put together indicates to us that these residues are essential for manganese transport by this transporter. So there's a bit of a summary of all that. So here's the PsaA, the solute binding protein. It delivers manganese to the transporter. The manganese needs to go through this pinch-off point. Once it goes through, it's prevented from refluxing back through the transporter. We have these residues here that coordinate the solute as it goes through. As you can see here, this is the aspartate and the histidine I mentioned before, and this provides a driving force for translocation through the transporter. At the same time, ATP hydrolysis to ADP causes a switch to the inward open conformation, and then the solute can pass through into the cytoplasm. So this is our model at the moment, but we have more work to do. So what we'd like to understand is how the PsaA interacts with the rest of the transporter, whether we can determine structures of other conformational states in the pathway of translocation, and importantly, that we can identify inhibitors of manganese transport. So to that end, Tess Malcolm in my lab has been collaborating with Mohsen and Isabel to try and see if we can study PsaBC by cryo-EM. And this is where we've got to so far. This is the map as it stands at the moment, which is at around 3.6 angstroms resolution, I believe. And so we're working toward establishing conditions where we can look at this transporter not only in crystals, but also via cryo-EM. We've also used a negative stain TEM to look at the complexes with those 8 fabs that I mentioned that Soawata provided us with. We were pretty keen to see whether we could identify fabs that bound to the membrane, the top of the membrane domain that might block the interaction with PsaA. Unfortunately, all the fabs actually bind to the nucleotide binding domain, which is not surprising, I guess, because that would be the part that was protruding most from the proteoliposomes when the fabs were generated. But you can see these TEM classes replicate the crystal structures really well. This is the one I showed you before. We've been able to solve another structure of a fab PsaBC complex, and it matches quite nicely with the TEM images. So in addition to trying to do all these things with PsaBC, we're trying to structurally characterize these other 2 proteins. Shadi Maghul is working on the CZCD, and Pramod Subedi is working on the zinc transporter. So at that point, I'd like to finish off and acknowledge my collaborators, particularly Chris McDevitt, who we've been working very closely with for a number of years on this project, and Stephanie Neville, who is a senior postdoc in his lab. Jenny Herham was in my lab for 2 years, and she did a lot of the very hard work solving the structure. The fabs came from Soawata's lab. Megan O'Mara did the MD, and Isabella Mosen have been collaborating with us on the cryo-EM. We're funded by the ARC and NH and MRC, and all the diffraction data were collected at the SSRL. As I've got 1 minute left, I wanted to do a bit of a pitch. I'm the president of the Society of Crystallographers in Australia and New Zealand, SCANS. This is not just a crystallography society. We actually represent all fields of science that look at the structure of matter, so that includes cryo-EM. If members, and particularly student members, have access to very generous bursaries to attend conferences called the Maslin Awards. So it's worth being a member. We have a conference coming up in December at Bendigo, and there will definitely be talks on cryo-EM. And that's all leading to the major international congress of the International Union of Crystallography, which will be held in Melbourne next year. And again, SCANS will be providing bursaries for students to attend that meeting. So it's well worth being a member. And on that note, I thank you for your attention.
Thank you, Professor, for that amazing talk and for solving these amazing structures. So I guess that we could now open the seminar for questions from our audience. So please, if you have any questions for our speaker, you could use the raise your hand function, or you could just type in the questions in the chat box. So I see questions here. Joy, go ahead if you want to unmute yourself and ask.
So was that me, Joe?
Joe, yeah.
Great. Sorry, I'm on my phone. My computer died. Thanks for the great talk, Megan. Great to see all the amazing work on ABC transporters, which I've started working on. But the thing I wanted to ask was, in David's lab, we were always working on proteoliposomes, and I never understood, is there like a topology preference in proteoliposomes? Do you expect the physiological topology to be recapitulated in the proteoliposomes, or is it like a 50-50 of the sort of inward and outward orientations?
Yeah, so the theory is it's 50-50. So we would expect when we put PsaBC into proteoliposomes that half of the molecules would have the nucleotide binding domain internal, and half would be external. I think in Chris's lab, they've been able to establish that by incorporating the GFP tagged protein into proteoliposomes and then trimming it, basically cleaving off the GFP and seeing how much fluorescence is left. And they find about half is left, which indicates about half of the protein goes in with the GFP internalized. But I imagine for some membrane proteins, there is a bit of a preference. And so I wonder whether the fact we see fabs against only the nucleotide binding domain, whether that's due to the fact it protrudes so clearly from the proteolypisome, or it's also due to, I guess, a preference for its orientation in the proteolypisome. It's a bit hard to tell, to be honest. Does that answer your question?
Seems like. So we could move forward to Isabel. Isabel, would you like to unmute?
Thanks a lot, Megan. That was a really great talk. I've got one general question and one more specific question. So you said there was some mechanism of resistance occurring. Do you know where is what is mutated? What causes the resistance? Is that in any mutation around the transporters?
Resistance to zinc, you mean, or?
All the, you said there's an antibiotic now not necessarily work, and there's mechanism of resistance occurring. Have this mechanism, the mode of action, be determined? Do we know where the mutations are? Or?
So if you mean generally antibiotic resistance, they become, so one resistance I'm familiar with is penicillin resistance, and that would be down to the beta-lactamase type resistance.
Yeah.
But I imagine, I haven't gone into it in great detail, I imagine there's efflux transporters as well, but the one I'm most familiar with is that sort of penicillin resistance, the typical type,
And so the more specific question, so, you said you need to have ATP hydrolysis to occur. So normally ATP hydrolysis will also require some carrying ions, so magnesium or manganese. So what is the impact? So when you have a, influx of manganese, so how much manganese that is going through is important for the hydrolysis of the ATP, if you see what I mean,
Yeah, that's a really good question. So Chris has done, we've measured the ATPase activity of the transporter in proteoliposomes. In that case, we use magnesium and ATP for that. But we also use a manganese-loaded PsaA to prompt the activity. Interestingly, the ATPase activity seems completely decoupled from the solute binding protein. So normally when you add a substrate-loaded solute binding protein, the ATPase activity goes through the roof, essentially. In this case, we see very little change, which is really perplexing. I hadn't thought about whether that's due to manganese binding in conjunction with ATP. Usually, if you use manganese in place of magnesium in other ATPases, it actually boosts the activity. So I'm not sure about that. We're wondering whether it's the fact that these nucleotide binding domains are so offset that actually it's really inhibiting the ATPase activity. And that sort of makes sense to us because in contrast to other nutrients that need to be brought into cells, manganese, once it comes in, you can't get rid of it actually unless you efflux it out via another transporter. It's not consumed like heme would be or other sugars, for example. And so perhaps the transport is very, very slow because it needs to be very, very tightly controlled how much manganese is brought in.
Yeah, thanks a lot.
Yeah.
Okay, and we will move to Jack. Jack, would you like to unmute yourself and ask your question?
Hi there, can you hear me okay?
Yeah.
That's awesome. First of all, thank you for a great talk. That was fantastic. So I just had a quick question, I suppose, about the PSA interaction with the integral membrane parts. And I know you touched on this is something you're still working on. But I'm curious with those sort of related families, the BTU and the H1 with the full extracellular helix, do you notice any kind of post-translational modifications that sit sort of along that ECD or any sort of homology between PsaA and the other sort of solute binding proteins that might indicate something of an interaction on how those sort of come together?
What we do know, what we know about for example, the vitamin B12 class is that generally there's glutamate residues on the bottom of the PSA of the solute binding protein. And then there's arginine residues on the top of the membrane domain, and these form salt bridges when the two things come together, and that helps it open and release the cargo. None of those residues exist on the top of PsaC. So we had all these great ideas about, oh, it'll be so obvious and we can make mutants and we'll work it all out. But actually, it's very, very difficult to find residues that would be comparable and basically mean that these two things interact by a similar mechanism. Megan O'Mara has done some docking, and that indicates she thinks that lipid actually might play a pretty major role in mediating this interaction. But she certainly can't, as I said, there's no arginines on the top of this. There were one lysine on each of these little helices, and we thought we worked it out, and it was the lysines. So we mutated those and also even chopped off the helices altogether, and that made no difference. So at this point, we have no way of knowing how these two things come together. So we really need structures actually to work it out.
Right. Okay. Can I also just ask, the PsaA, is that anchored to the membrane in some way, or is it just sort of free floating just outside?
Yeah. Yeah, it has a membrane anchor. Yeah.
Okay. All right. All right. Thank you so much.
Okay, I see Isabel again.
Yeah, it's following on Jack question. So is there a conformational change of the PsaA upon ligand or upon zinc or binding or metal binding?
Yeah, so that was that movie I showed before. So it's sort of open when it's metal free, and it closes in as residues come in and coordinate the metal.
And is this mechanism, is that pH dependent? Would it be the binding of metal be pH dependent? So the code, because you would think so coordination, I don't know.
It could be, I'm not sure if Chris has measured that. I would imagine since there's histamine residues involved in that coordination, that there would be some pH dependency, but I'm not sure if he's measured that, to be honest. Yeah.
And so you have no indication that the transmembrane part will have any conformational changes?
Well, we expect it to be that way when the solute binding protein docks, but we just don't know where the interface is between the two proteins at the moment. So that's what we really need. So we've made some attempts to co-crystallize the two proteins together, and we haven't been able to do that yet. So we're hoping the cryo-EM might help us in this regard.
Do you think there's a component missing or something else linking?
Well, it's possible that it needs lipid, and that might be why it's so difficult to make the complex. And so perhaps using SMELPs or nanodiscs or something like that might be the way to go. So this is, we've been working in detergents until now, and it's possible we need to do something different, I think.
Okay, yeah, thanks a lot.
Do we have any other question for our speaker? Let's see. Okay, maybe I could ask a question. So first of all, thank you for including, some more information for our students. So thank you on behalf of the students here on the seminar. And I have a question. So do Fabs bind in the same part always on the protein, like always on the nucleotide binding side?
The Fabs that we have do. And as I said, I think there could be a couple of reasons, but one of the reasons would be, I think, because the nucleotide binding domain protrudes from the proteoliposome. Whereas, sorry, let's see if I can go up. So if you can imagine this is the proteoliposome, we don't have much structure of the transmembrane domain outside the proteoliposome for antibodies to recognize. Mm-hmm. And so this could be why we're getting all the Fabs against this part here. We do have one idea of how to solve that. We're going to work in collaboration with Simon Newstead and basically put a tag on here so that we can orientate the protein and then pan psi bodies that recognize the top. So that's our next step. This will be a collaboration with Simon Newstead from Oxford University. Yeah.
Good. And that answered my next question, like, where do you see this going?
Yeah.
Nice.
Yeah. So we really need to find something that will bind to the top, because then we can test this sort of proof of concept, whether if we can interfere with the interaction with PsaA, whether we can inhibit manganese transport. And so this is what we're trying to do, identify binders that basically interact at the top here.
really nice. Thank you so much. Do we have any other questions from the audience? You could use the raise your hand function, or you could type your question in the chat box and we can address it afterwards, or I could read it for you if you don't feel comfortable. Like, okay, it seems that we don't have any other questions. So on behalf of everyone here today, organizers, audience, and myself, I'd like to thank you, Megan, for joining us and sharing your amazing research. And thanks to everyone who joined our seminar today. We hope to see you all in the next one.
Thanks very much for having me.